for the restarting of our Vision Seminar series. We have a group in person here, and then also it looks like quite a few people online. We'll try and make sure that we repeat any questions that are coming from the audience here in person so that the folks on Zoom can hear. And then we'll also be monitoring the chat. If anybody that's on Zoom has a question, please feel free to weigh in. So with that, it's my pleasure to welcome Jesse Schallek here to the University of Pennsylvania for our Vision Seminar series. Dr. Schallek is an assistant professor at the University of Rochester, and we're really excited to have him talk with us today. His research in imaging with adaptive optics, the visual system, and blood flow is just totally stellar. So I'm really excited to see his images and hear about his science today. So with that, Jesse, welcome.
All right, I think we're online. And so thanks to in particular, Jessica, for the invitation to come to Penn. This has been great reading for so many years about the people that do the fantastic research at the center. And even though I've read about it for many years, it's the first time being on campus. So this is a nice way to come back out of the virtual world back into in-person meetings, and hope that this is the very beginning of many meetings to come. So a quick introduction of myself. I've been at the University of Rochester since 2010. And after a short stint of doing some postdoctoral work in the adaptive optics imaging group there, I began faculty life in 2015. And over the past 7 years or so, the major focus of my research group has been to apply advanced imaging technologies to understand the vascular perfusion of the back of the living eye. And what we've, the central tenet of what it is that we're after is turning a tool that was once an anatomical tool to understand the anatomy and mosaics of photoreceptors. This is adaptive optics technology, to turn that tool into something that could be more quantitative and understand the structural and functional perfusion of the back of the living eye. So one thing that doesn't need much introduction, I think, to this audience, let's see if I can pause these guys, is what the back of the eye looks like. And if you're a visual neuroscientist, you think of the sheet at the back of the eye is filled with neurons and glia. But if you're a vascular biologist, you also pay strict attention to the vascular perfusion of the back of the eye. In the human eye, there are two major circulation pools that supply the metabolites, oxygen, and all of the waste product removal to the back of the healthy eye. We're going to go over some of the anatomy because that warrants the discussion of this talk, what those different structures are. So as the light comes in through the back of the eye, we know that the image is focused on the back portion of the retina. And if you look at an image like this, this is the optic nerve. And again, this is the point of entry of all of the vessels that come into the eye. It's also the point where all of the visual information leaves the eye en route to higher brain structures. When an ophthalmologist looks at this image, one of the first things they're going to look for is how do the vessels look in the back of the eye? They're going to look at the anatomy of that eye to see if the patient is in good health. That reddish tinge that you see, that's macular pigment plus the hemoglobin of this highly vascularized tissue. So some technologies over the years have been able to now give basically an in vivo optical biopsy of not only the neural retinal layers that you see in cross section, but also understand the vascular perfusion of the back of the eye. So this is optical coherence tomography that many of you are familiar with. And this is data from my own eye, which shows that you have these two circulation pools. On the surface, hopefully everyone can see my mouse cursor. On the surface of the eye, we have single vessels. Let's see. We can get any feedback, and hopefully the animations are playing over Zoom. I'll keep going. If there's still problems, stop me again, Jessica. Okay, great. We're going to hide email as it comes through. When we have this vascular perfusion to the back of the healthy eye, this is another schematic. This is not data, but this is an illustration or a cartoon of what would happen if you took a cross-section of the back of the eye. This cross-section allows us to see that all of the vascular supply to the eye comes through the central retinal artery, and then all the return is returned through the central retinal vein. This is for the retinal circulation only. We also have this dense vascular pool behind the photoreceptors and RPE cells, and that's the choroidal circulation. So again, the neural retina in between, this dense vascular pool of the choroid that resides behind the photoreceptors, and this supplies about 80% of the metabolites to the neural retina. And the focus of my research is to understand the retinal circulation. This is the circulation pool that resides on the surface of the retina and interdigitates in more superficial layers of the neural retina. We're going to focus most of this talk on just the retinal circulation. We're going to ignore the choroid for today. That's a topic for another talk, but equally important. The reason that we are focused on retinal circulation is because it resides right on the surface of the retina, and it provides an optical way that we can assess individual blood cells in the vascular perfusion. It's also a very interesting organ at the vascular perfusion level because the eye and the visual system has to deal with this conundrum. On the one hand, neurons are incredibly metabolically expensive. They need a lot of energy in order to function properly. But at the same time, that vascular bed can occlude photons that reach the photoreceptors. So if you're a visual animal, like most primates are, you care about high visual acuity information. And most of you know that we have a foveal pit. What some of you may or may not know is that the foveal pit is generally devoid of having blood vessels. This is what's called the foveal avascular zone. And so we can clearly see that there's a trade-off between a highly vascularized tissue and preserving high visual acuity information. So I'm going to start today's talk by reminding all of you that the stuff that's pumping through our bodies, through all of our waking moments, from the day that we're born to the time that our heart beats its last beat, is not just this red stuff that we call blood, but blood is actually cellular in nature, right? We have about 25 to 35 trillion blood cells in our circulatory system. And then hopefully this needs no introduction, but this is a scanning electron micrograph of the familiar red blood cell, a white blood cell and sort of pseudocolored in blue, and an activated platelet in yellow. And the goal of my research might have seemed like science fiction maybe 20 years ago, but what we're after is to be able to understand and visualize these cells inside the living body. Normally, a hematologist would want to take a little drop of blood and you put these under a microscope in order to study these cells. But we're after detailed visualization of these single cells. And the eye just happens to be a fantastic place that you can look for these cells. So how are we going to do that? One way, if you're a first-year bioengineering student or neuroscience student, might suggest that you go into the back of the eye. This is what most clinicians would see if they took a picture of a healthy young eye. Optic nerve, and what you see at the center of that box is a macular region with the fovea right at the center. And your strategy would be to optically magnify this image to the point where you got down to the level of single photoreceptors. That's okay. And so when we get down to this scale, this is just 1/10 of a millimeter. It's at this magnification that we should be able to see those blood cells. But in fact, you see nothing. You see basically a blurry mess. And that's because the front of the eye have what we call aberrations. This is what you might be familiar with, defocus and astigmatism with spectacle correction, but also what we call high order aberrations, which all collectively blur and distort the image. So this is bad news for the pursuit of imaging those blood cells inside the living eye. Some of you might be familiar that we're using a technology called adaptive optics. And when you apply adaptive optics correction, we can see individual capillaries now in the back of the eye. And if you combine this with video rate mode, we can see the movement of those single blood cells moving through the capillary network. And I like superimposing this image on top of what you saw from adaptive optics, which is to scale a human hair to remind us that we really do need this diffraction limited adaptive optics imaging if we're to ever visualize single blood cells inside the living body. Even though we were very excited, is there a way, Jessica, to hide? Even though we're very excited about 10 to 15 years ago to be able to produce images that you see in the center of that slide, it still lacked a lot of the detail of those blood cells. And our group, along with a number of others, have been able to revolutionize the adaptive optics imaging so that we can see now and study individual blood cells inside these tiniest of capillaries. This talk will not be about adaptive optics because that would take an entire seminar to go over it, but it does warrant about a minute of explanation. This is a highly simplified schematic of how adaptive optics correction works for the eye. This is an animation provided by David Williams. In an adaptive optics system, a single point of light is shown into the back of the eye. And as that wavefront comes out, if I pause it right here, hopefully you'll see that that wavefront is a little bit distorted. That's what distorts or blurs the image that we're trying to get of that high resolution picture of the back of the eye. That's bad for imaging. That point of light is reflected through and arrives at, a wavefront sensor. Let's see if we can play this again. The wavefront sensor chops up that wavefront into little tiny pieces and allows us to calculate what the aberration state of the eye is. And through a number of different iterations, that wavefront sensor then talks to a computer, which sends a control signal to this element, this deformable mirror, which has a number of different pistons and actuators on it to bend and correct for the aberrations of that light coming out of the eye. So we measure the aberrations and then we correct for them. This iterates many times per second, about 13 times per second are these corrections measured and applied. Finally, then you'll see the light comes into the eye after we have the aberrations corrected. We're able now to use a different wavelength of light so that we can image the structures of the back of the eye. As this light now comes out of the eye, we're able to get a distortion-free image or an aberration-free image of very detailed structures of the back of the eye. And you might be familiar with a number of the structures that can be imaged inside an eye. And perhaps this is one of the most famous images, imaging individual rods and cones. This is data from Alf Dubra and Joe Carroll, which shows the photoreceptor mosaic in the back of the eye. And in fact, adaptive optics has had a great history in being able to do some of the first imaging of these photoreceptors in the back of the living eye. What I'm here to tell you today is that adaptive optics has really matured beyond just a tool for imaging photoreceptors into a really powerful tool that we can assess many other structures in the back of the eye. If we look at this cartoon here, this is a highly schematized version of the cross-section of the neural retina. If you were to image with traditional adaptive optics imaging, you might be able to see very detailed structures of things like capillaries, the nerve fiber bundles, and the photoreceptors. But what you definitely see is lacking in this image are the visualization of most of the other cell classes in the retina. We care a lot about those because those go wrong in disease. We've then combined over the past 7 years or so a new modality called phase contrast, which allows us to get very detailed images of translucent cells in the retina. And this is most neurons in the retina, right? So light has to travel through these in order to get to the photoreceptors. And millions of years of biology have made these cells clear, which is good for vision, but very challenging to image them. So we've combined this phase contrast so we can image things like ganglion cells, blood cells, horizontal cells, the somas of photoreceptors, and other structures. And all the data that I'll be presenting today is not from the human retina, but instead from this laboratory animal. This is the laboratory mouse, in which we built a custom adaptive optics camera that's designed to image the tiny optics of the mouse eye. And of course, the mouse is a fantastic model to not only recapitulate aspects of human disease, but there's also a rich transgenic catalog where you can basically express any protein that you'd like in the eye, tag it with a fluorophore, and now get sparse labeling of specific cell populations inside the back of a living eye with resolution similar to what you get with some of the best light microscopes in the world. So today's talk is going to elaborate on 3 separate themes. The central theme is imaging blood cells and their perfusion in the back of the eye. This central theme is going to be broken down into 3 acts. And first act is imaging the tiniest of capillaries and seeing single blood cells at these tiniest of capillaries. This is, of course, important for understanding microvascular disease because most microvascular disease originates at the level of capillaries. In order for us to understand what's going on, we'd like to be able to see those capillaries. The second act is going to describe how it is that we can measure the blood cell speed in the biggest of vessels. Even though you can see those big vessels, doesn't mean that you can measure the speed of those blood cells in the big vessels. And we're going to go over some software and technology that we've built that enables that study. And the last part of today's talk is going to go over what happens to imaging blood cells when they leave the blood vessels. Well-behaving blood cells should remain within the vasculature. But when you have this response to inflammation, those single blood cells actually leave the vessels and they turn into inflammatory propagators inside the diseased retina. All the data that I'll be showing is from the laboratory mouse. If you haven't been very close to your local house mouse, this is what our preparation looks like. So the mouse is asleep on the table. You might see a nose cone that the animal is breathing, isoflurane anesthesia, and we can hold this stable preparation for about 2 hours. The mouse eye is also dilated, which gives us this nice, beautiful, round, dilated pupil, which gives a very large numerical aperture, which allows us to go right in and be able to study the vascular perfusion of this sleeping animal. There are a number of notable differences between the mouse eye and the human. I won't get into all those, but basically mice don't have a fovea. Mice don't have all of the same photoreceptor types that humans have, but otherwise what I want you to remember is that the mouse retina looks in more ways similar to the human retina than different. Let's turn our first part of this talk to focus on the capillaries. These are, of course, microscopic. These are about, you could stack anywhere 10 to 20 of these across the thickness of a human hair. And this is where the adaptive optics imaging allows us to see the movement of those cells. So in 2010, this was sort of the current state of the art, combining adaptive optics plus video rate mode so you could see the twinkling or movement of those blood cells inside the vascular network. But just recently, we've been able to combine adaptive optics plus phase contrast to allow us to provide greater detail of these translucent cells. So just seeing the twinkling movement of blood cells is interesting, but we can gain far more information by looking at individual rheology and shape of blood cells. Let me pause this image right here. And in any given frame, hopefully you can see where my mouse cursor is moving, the shape and deformation of a single red blood cell. This is captured at 25 frames per second. And this has really changed our ability to be able to visualize how the blood cells are moving through this vascular network. So I play this video, pay attention to where my mouse cursor is. You'll also notice that specific vessels perfuse and then stop their perfusion. This capillary right here is actually arrested perfusion. And about 10 to 15 years ago, this was thought to be a very rare event in the mammalian retina, but we're finding that it's far more common than had previously been appreciated. In order for us to study the detail of those blood cells, we don't just need resolution and contrast, but we also need a third ingredient. We need a very fast camera in order to visualize those blood cells. And so our strategy was borrowed from Steve Burns in Indiana, and actually Ray Warner's in the audience here today and participate in a lot of that work, where we take a single beam of light and we scan it very quickly across a single vessel. So in these AOSLO systems, we have a raster pattern, basically a flying spot that scans an image. In line scan configuration, we're just taking that single beam of light and scanning it very quickly across a single vessel of interest. And when you do this and you scan 15,000 times per second, or double that at 30,000 scans per second, what you have is a literal ticker tape record of all of the blood cells that pass that beam per unit time. So here with fast camera capture, we can image the rheology of individual red blood cells as it moves through this capillary network. Why is this important for clinical medicine? A number of techniques have been able to image blood flow in bigger vessels. Fewer still been able to understand what happens in capillaries, and almost none tell us about the packing density of those erythrocytes in the tiniest of capillaries. Why is this important in disease? If you saw that a capillary was perfused, and you might know that from a fluorescein angiogram, being able to see whether or not the capillaries are perfused, you'd have effectively no information of how the blood cells are moving through that capillary. And now with adaptive optics, we can now appreciate a rich level of detail about just how those blood cells are packing within those capillaries. What's remarkable about this image is these are 5 different capillaries of almost equivalent diameter in this mouse. And yet, even though they're all perfused and they're all the same diameter, the packing density of those erythrocytes is really different in each one of those capillaries. And these are all, as far as we know, healthy capillaries. This is important for microvascular disease because you could imagine clots forming or different imbalances happening when you see these different packing densities of these blood cells. The image at left, or all those images at left, that's in vivo data, and the images at right are borrowed from the paper from McWhorter et al., which show the finite element analysis of the lowest energy state of the packing density of those red blood cells in capillaries. So here the in vivo data is matching what the theoretical models are of how these blood cells should be packing within capillaries. So it's a nice marriage of engineering and science realized in the same image. If you looked at that last example, there are a number of different conditions where we see the blood cells stop. And we in a number of groups have shown this, but the video that you see at left has been sped up by 10-fold. You can see the formation of a clot, almost like the smallest stroke that you could see inside the central nervous system. And then it reperfuses. And now we're in a position to answer why those clots are forming and maybe in the future, how to prevent or mitigate those kinds of events, because stopped blood flow is very bad for central nervous system neurons. The image at right, I'm going to pause this video at just the right point in time, right here. Hopefully you can see, I'm going to draw your attention here. You're going to see the movement of just one cell that sluggishly moves through this capillary. And this reminds us that it's not just red blood cells that move through that capillary, but sticky and less deformable white blood cells also have to flow through the same capillary network. So we're very excited about this because now that we can see it, we can better understand and hopefully mitigate these kinds of events inside the healthy perfusion. We're also getting a better appreciation of just how often this stuff is happening inside the healthy retina, which has to be categorized first before we can understand this and apply it to disease. All right, so let's take a pause. That's Act 1. That's understanding adaptive optics and seeing capillaries inside blood cells inside capillaries. The second act is going to shift our attention away from the capillaries and now study some of the biggest vessels. So as I told you at the first part of this talk, that all of the supply to the retinal circulation is supplied by the central retinal artery and the big retinal veins that drain all of that into the optic nerve head. You might ask the question, why do we need adaptive optics at all to study these big blood vessels? I can see them just fine in my ophthalmoscope. And we make the argument here that adaptive optics is still very important because maybe you can see the blood vessel itself, but you can't see the blood cells within without the adaptive optics assistance. So for this part of the talk, I'm going to tell you about how we've developed tools which allow us to see the blood cells inside these biggest of vessels. So the image you see here, this is raw data that's captured at 25 frames per second inside a living mouse. And each one of those little streaks that you see, that's the passage of a single erythrocyte. Our strategy is very similar to increase our temporal resolution. Now we're taking a single beam of light and scanning it very quickly across this one big vessel. And when you scan across a big vessel in this way, the data that you produce looks like this. Let me unpack this for just a second. The distance across that vessel is represented along the y-axis. This is the space dimension. And every single line that we capture is presented across the x-axis. We call this a space-time image. Okay, so it's not XY image, but an ST image. And each one of those diagonal streaks that you see at the image at right, that's the passage of just one erythrocyte as it passes by the beam. We know the frame rate, we know the spatial resolution and pixel pitch of the instrument, and therefore we have the basic ingredients to make detailed measurements of single cell velocity inside these biggest of vessels. This here. Yeah. The question that came from the audience is what is the sort of undulation or the pattern that we see beside this vessel? This is actually the nerve fiber bundles of the mouse retina. So what I'm not showing you is up to this location up here. If you continue to follow this vessel, you'll get to the optic disc, and the optical, the retinal nerve fiber layers create these bundles which project in similar ways to the vessel. Great question. So these are tough images to understand and appreciate. And what we've done here is we've created a simulation of what it is that we see in these space-time images, because it's not, at least for me and most people that I speak to, not intuitive. So working with our optical engineer and computer engineer whiz, Keith Parkins, we've developed a simulation of what it is that we're seeing in these space-time images. So we've created 3 classes of blood cells in the simulation: red cells moving very fast. You'll see that at the central column, medium cells in green, and slow cells in blue. And we're simulating what happens when we have the single beam of light represented by that yellow trace, and we're creating the space-time image at right. So what I want you to pay attention to is the fast-moving cells have a slope which is very steep. The slowest moving cells, which are at the boundary condition, are at the very edge of this image. And hopefully you can see in the simulation that there is a pixel pitch or a slope pitch to each one of these cells, which represents sort of a sigmoid function. This is the simulation at top and the real data at bottom. And hopefully your eyes can see there's sort of the sigmoid pattern, right? The boundary condition in which blood cells are moving the slowest represents the slowest or shallowest slope, and the fastest moving cells are moving in the central column. So it fits what our expectations are of how blood or particulate matter should be flowing through these big vessels. This is the only point in the talk where I'm going to delve into a little bit of the math, and I promise I'll keep it very light. But our strategy is, this is really great that we can visualize these cells, but we need to make this accessible to people that might want to understand vascular perfusion. So I'm showing you this very unique image that you see in the upper left, a space-time image, but this doesn't carry any information for someone who might like to use an instrument like this. So our objective through engineering is to make this simple. Let's focus in on just one little location within the space-time image. This little box that you see magnified here, that's a single erythrocyte at the center of that image. Our task now is to calculate the velocity of just that one blood cell and then expand that or scale that up to all positions within the image. And we don't want a sea of graduate students doing point and click in order to measure the velocity or slope of those cells. We want a computer to do this. And we could do this in a number of different ways, but the way that we've chosen is something borrowed from computed tomography. Something called the CAT scan or computer-aided tomography uses an algorithm called the Radon algorithm. And again, I won't go into too much of the detail, but imagine if we took that image and we created a column-wise summation of the pixel intensity of that circle. And then we rotated the image a little bit and iterated that many times. What you'd produce is an image that you see at right, and this is called the radon image or sinugram. That sinugram contains with it an area that has highest contrast, and that highest contrast in the sinugram corresponds to the rotation angle that we rotated that image so that all the streaks are now vertical. If we know that vertical orientation relative to the actual orientation of those streaks, now the computer knows what the slope is of those cells. This is what a sinugram looks like. What we're looking for, or what the computer looks for, is the area of maximum intensity variation within the sinugram. And we produce a plot that looks like this. The computer is now solved for an angle which is approximately 85 degrees. That 85 degrees now encodes the velocity of just that cell. Okay, so we can now compute that velocity into an absolute number. That's 22 millimeters per second. So now we know that one cell that we measured is moving 22 millimeters per second. That's the basic ingredients of this strategy. And of course, we can scale that up. And now the computer goes in and evaluates all the different locations within an image, right? Again, without user input or without a human having to manually do this. And it basically creates a signal to noise threshold, which says, I'm going to analyze all portions in the image. I don't care if I get a good solution or a bad solution, I'm going to report a result. But then we need to scale those reported results to what's believable and what's not. And this is where we create a very crude signal to noise characteristic. Any location that has a very strong peak, that means that there is a very good, strong signal, which corresponds to the velocity of blood cells. Anything that creates a gray peak, or basically low variance in the sinugram, contains very low signal to noise. So here we're able to gate all locations with just 2 colors that says, believe the data or don't believe the data. And it fits very well with what our eyes see. Our final objective, now sparing you the details of any of the math, is to take an image that looks like this. If we were to give this to a clinician, they'd say, well, it's great that you can image the streaky stuff, but what does this mean for blood flow? We need to take an image like this, and through Keith Parkins, we've now created an algorithm which can create the analysis in real time of over 100,000 measurements per second. Again, this is about 30,000 scans per second, so it's a lot of data. We've combined this with GPU architecture, which allows us to measure the local velocity of hundreds of thousands of these blood cells over time. What does this allow us to do? It's not just a parlor trick. Now with an AOSLO, we can now measure the biggest vessel in that organ all the way down to the smallest vessel in that organ. Okay, so think about blood speed in your aorta versus the capillaries in your fingers or toes. Very different size, different scale. Here we're able to image the entire organ from the biggest vessel to the smallest vessel in the same instrument. The reason that this is important, and this is a publication that's now in press, is that the relationship between the diameter and the passing blood cells is actually very poorly characterized. If you think about basic plumbing, you might scale down that you know that smaller diameter is going to give rise to smaller blood flow, but that relationship really breaks down due to the non-Newtonian flow of single blood cells. And at the capillary level, again, where disease originates, there's very poor relationship between the diameter of the capillary and how many blood cells are moving through that capillary. And this underscores the importance of doing the direct empirical measurement. We can't model this and understand how vascular perfusion is working. We need to be able to image it in order to understand what's going on. Why is this relationship so poor? One of the things that we found through imaging these single blood cells is that local hematocrit is variable. So if I took a drop of anyone's blood in this audience and we sent it for blood work, one of the numbers that you get is the ratio of plasma to red blood cells, the hematocrit. That's true for all the blood in the body. But if you look capillary by capillary, some blood cells pack in very densely, like this condition, whereas other capillaries have very sparse blood cells. And this is due to the Ferrius-Lindqvist effect. And I won't get into the biomechanics of this, but basically some capillaries have more plasma than others. This is really important for understanding single neuron metabolomics. I did mention all the data that I'll be showing today is from the mouse, but just to whet your appetite that this is translatable. The data that you see from a mouse is at top, human data. This is data from my eye at bottom. And you see a number of notable differences. One is just very simple heart rate differences between mouse and man, very different. But basically the central tenets are the same, that you can image and quantify velocity in now not only vessels of all size, but vessels in different species as well. All right, for the balance of today's talk, I want to focus in on some of the data that we're most excited about. The first two acts of this talk, we talked about how blood cells are moving through capillaries and biggest of vessels. But again, blood cells are supposed to stay inside the vascular system in conditions of health. Anytime you have a diseased eye, especially microvascular disease, blood cells actually come out of the vessels, right? This can happen in a number of different ways. One is a bleed, right? And so you could have a rupture, for example, in an aneurysm, or a leaky blood-brain barrier that you see in a number of different diseases, and blood actually leaks out. But there's another interesting biologically inspired mechanism by which blood cells leave those blood vessels. And that's through extravasation. That's single immune cells that purposefully move through the vessel wall that get into an inflamed neural tissue. And that's what we're going to cover in the last part of today's talk. This can be appreciated in a number of retinal degenerative disease. I'm just going to select one here today. This is diabetic retinopathy, which is a vascular disease which impacts many millions of Americans and people worldwide. It's primarily thought of as a vascular associated disease, and an ophthalmologist will look at an image that you see in the center there and notice things like arterial narrowing, venous beating, nerve fiber hemorrhages, cotton wool spots, preretinal hemorrhages, hard exudates, all features of an abnormal vascular perfusion. But these are just structural components of what's going on in diabetic retinopathy. The other tool more recently that they have at their disposal is to take a cross-section. So if we take this line here, this is a nice review by David Antonetti. We take a cross-section through the macula. In the diabetic retina, often you'll see something called diabetic macular edema, a thickening or swelling in the diabetic macula. And this is really what causes a lot of vision loss. But here again, we're sort of lacking in the tools and understanding what's going on to create this inflammation in the diabetic eye. Is it just leaks? Certainly more to it than just leaky vessels and poor blood retinal barrier. And this is where we think adaptive optics might be able to help us. What comes next is a collaboration with a fantastic clinician scientist who came to work in my laboratory for 3 months, and we're able to accomplish quite a bit. This is Colin Chu, who's at the University of Bristol. And for 3 months, just a couple of years ago, Colin came to Rochester to do immune cell imaging inside the eye. And in order for us to image immune cells, Colin brought with him a model called endotoxin-induced uveitis. Uveitis is basically a swelling of the uvea, or the lining or the back portion of the eye. And in order to model this in a mouse, we can do a very simple thing. We place a single injection into the vitreous of the globe of the eye. And this injection is a dead bacterial cell wall. It's called lipopolysaccharide. It inflames the retina and makes the retina irritated without actually touching the retina. And Colin in 2016 showed in other mice that when you're imaging with OCT and you look at the healthy retina at top, you get this nice flat retinal laminar organization. But within just a day or so of that injection of LPS, that you get a very different looking OCT image, right? So this is the vitreous humor. Here we have the neural retina. And he said, you know, boy, when we talk about this work, we can see all of this debris in here. And we think that these are single cells, but of course we don't really know. It could be just a debris field, dead stuff, or particulate matter, or those could be cells. And then he would also sort of point in the center of the image and say, well, there's also hyperreflectant structures in there, but who knows what those are? Are those inflamed ganglion cells? Are they dilated capillaries? Are they immune cells? So this was a perfect project for adaptive optics, in my opinion. And hopefully the data that I'll show you convinces you that it was a good project. Here we're taking a single vessel from a mouse that's otherwise healthy. This is a retinal venule. Again, I'll sort of orient you here. The optic disc would be right here in this image. You can see a big vessel, healthy capillary network, and then some of the questions in the audience, you might be able to see tiny structures in here that are indicative of ganglion cells and the nerve fiber bundles that pass through. This is what the healthy retina should look like. When we make that single injection and inflame the retina, we get a very different appearance of the retina just 72 hours after we make that injection. And we were blown away initially by an image like this because we said, or we thought that these were immune cells. They certainly had the shape, the morphology, the size, the appearance of single immune cells. But how could we go the extra mile to confirm that these were in fact immune cells versus, for example, swollen glial cells or neurons that became inflamed? Next few slides hopefully will convince you that these are in fact immune cells. And the way that we can convince you are one of two ways. One is that we can label those cells with a fluorescent agent with known fluorophores. And again, the benefit there is that in mice, we can label these cells and inject them and understand how they're working. The other is a much more simplified approach. Just look at the kinetics of those cells. Most of the macroglia and neurons of the retina should not be moving around, but immune cells are very mobile. They're very motile, and we should be able to see them moving. So how could those cells be getting into the neural tissue? The image that I show at right is basically from high school biology 101, which shows the immunological process of extravasation of white blood cells. Normally, white blood cells are in free circulation within a vessel, but when they encounter an inflamed tissue, they start to roll along the vascular endothelium through slings and tethers, the selectins and integrins expressed on the surface of those cells in response to different chemokines and cytokines on the boundary of that vessel. And this is a cartoon, of course, of how those blood cells bind to the edge of the vessel. But what we're seeing in adaptive optics really is, by no stretch of the imagination, exactly what the cartoons show. These are now rolling single white blood cells along the vascular endothelium in response to this inflammatory response. And remarkably, this is happening just 6 hours after the retina gets inflamed. So this is one of the first steps of being able to see those immune cells. This would be a difficult experiment to do in a human, not impossible, but difficult. And here what we're doing in mice are we're labeling a subset of those white blood cells. These happen to be neutrophils, which are labeled with a fluorescent agent tied to Ly6G. And what I want to point your attention to is the image at top. You might see these binding cells at the top, and they co-localize with the fluorescent image at bottom. Let's see if I can play this again. So what I want your eyes to do is to recognize the image that you see at top, really co-localized with the rolling objects that we see with the fluorescent agent. So not only do we know that these are immune cells, but we can also label the subtype of immune cells, in this case, neutrophils, and know that those rolling cells, at least in this mouse, represent neutrophils, subpopulation of white blood cells. The last tool that we can throw at this is not just fluorescent labeling, but if we wanted this to be useful in humans, wouldn't it be great if we could just image these cells without any fluorescence at all, just to use that phase contrast technique that I told you about and to be able to visualize these cells without having to inject any fluorescent agents or cells into the human body. And this is our pursuit of this project. What I'm about to show you is this is an image of putative immune cells. And what I want you to pay attention to is the time ticker here in the upper left. This is a sped up image. It's a time-lapse image. If you just grabbed a couple of these images from the inflamed retina, you wouldn't see any movement at all because the cells are moving so incredibly slow. They're moving at a glacial pace. But when you register this data and speed up the playback, you look at how quickly that clock is running, you can see really the purposeful movement of those immune cells as it interacts with the vascular endothelium. This, we think, will be a game changer for understanding all inflammatory diseases of the eye. So we have hundreds of these kinds of videos, and you can spend hours just looking at these and sort of imagining the behaviors of individual blood cells. But I'm going to draw your attention to just two cells in this image to make a point. The first location that I want you to pay attention to is in this red circle. And if I time this just right and announce it, you'll see right here a single immune cell that was once within a vessel, right? That's a circulating immune cell that's inside the vessel. It leaves the vessel. And to our understanding, this is the first time that this has ever been seen. This is called extravasation or diapedesis. First time that this has been seen inside the living eye. This is exciting because this is the initiating step, which gets the immune cell into the tissue. Once that cell is there, then it can go about its business of phagocytosing, getting debris in the retina that associates with photoreceptor loss and a number of other cells that go away in retinal degenerative disease. But that cell also, once it's in the tissue, recruits other cells to come, right? It's the sentinel cell that starts signaling to all the other cells that there's a problem in that location, and it purposefully recruits more cells to the location. Let me show you one other location. This might not seem quite as interesting, but caught us by surprise. If you look at this green location, you actually see an immune cell come back into the vessel. And this might not seem like a big deal until you talk to an immunobiologist. And this really is never supposed to happen inside these tissues. In the central nervous system, it generally lacks what's called the lymphatic system. This is the superhighway for immune systems to interact with the tissue and then leave that tissue, go back to the lymph nodes or myeloid producing organs of the body and create specific immune response to different antigens that are expressed in the body. This is the nature of a number of autoimmune diseases that go awry. The reason that this is very important to see this in the eye is that the eye doesn't have a lymphatic system. It only has blood vessels, as far as current science understands. It would be very interesting if this is unique in this tissue, that immune cells are following within the vasculature, the blood flow system of the eye to both come into the tissue and remove themselves out of the eye. This would be very important to understand, not only in the eye, but in the brain as well, that operates in a similar way. This is now just sort of eye candy, a number of different sequences which show the different immune cell behaviors. This is all the same imaging modality, but now you can see just really different behaviors of these immune cells as they start interacting with the tissue. I'm gonna let this play a few different times and point out the different behaviors that we see here. In the first images that you see at the left, we see, cue this up, the rolling of these blood cells once they're inside the vessel. We can see single cells leave the vessel. You see here this extravasation. We can see the swarming of these cells. If you pay attention to this vessel, you can actually see blood cells moving or crawling along the vessel. If I pause this here, you'll actually see a very interesting behavior that we had never seen before. This is actually an immune cell that's moving upstream within a vessel. So it's actually exerting energy to move against the direction of flow inside the vessel. There's got to be a reason for that cell doing that. This is the purposeful movement of a single cell that's marching against the direction of flow. And then also a number of different interactions with native immune cells, microglia, with the vascular endothelium that recruits more immune cells into the tissue. The benefit, of course, of adaptive optics is that you can come back to the same tissue time and time again. It's non-invasive, right? You don't just have a single time point, but you can look at a time lapse of data, not only within a single session, but here we've imaged a normal, healthy baseline image in a mouse. We then inflame the retina through this LPS. And now we can return to the same location just 6 hours after the injection. And if you notice here, this is a venule, and you see the rolling of these cells right here. But in the arterial, which is right here, there's no rolling. This is a known behavior of immune cells, that they only tend to roll within venules and not in arterioles. Now let's bring that same mouse back 24 hours later. Same retina, same mouse, just a day later. And the appearance of immune cells, very different, and the motility is quite different as well. 72 hours later, 10 days later, 2 months later, this is what a clinician dreams of in the inflammatory response of the retina. Immune cells coming in to do their job, to clean up debris, and then leave the neural retina largely intact. Unfortunately, this is usually what goes wrong in a number of uveitic diseases in humans, is that the immune cells come in, they wreak havoc, and they start damaging otherwise healthy retina. So now that we can see this in a mouse, we're hoping to work with a number of different pharma applications to address how we escalate or suppress the immune response so it has a favorable outcome in the neural retina. Images are great, but what are we going to do with this? We have to image these big vessels. We have to count the cells. This is work by Guangping Feng, a bioengineering student in my group, and not only image and count the cells, but also track their behavior. So he's developed a deep learning network, which is able to identify and track individual immune cells with instant segmentation and track their motility as a new biomarker of how inflamed the tissue is. So here's a time sequence, baseline, no inflammation at left, all the way out through 10 days. And our first task is, how many immune cells are there? That's a basic question that we'd like answered. The way you could do that is you could take a whole bunch of users, naive and expert graders, and just say, how many cells are there? Time point 0, no cells, right? Very few in a healthy retina. After this LPS injection, an escalation between 6 and 24 hours later, and then by 72, basically very few immune cells again. All the blue data, that represents individual expert graders, counting hundreds of these images over many different mice. The purpose of Guangping's work is to now have a computer do the same assessment. And the goal isn't how many cells were in that location, just does the computer match what the humans find? And the severity scale that humans and this automated algorithm find show a very good match. The second thing that we want the computer to do is very difficult for a human to do, and that's to track the motion or behavior of these cells. So we're going to focus on just this one cell here. And here you can see Guangping's automated algorithm that's tracking the motility of this cell. This is over about a minute of time. Okay, so if you were to ask a human to do this, over hundreds of videos, many, many mice, this would be really challenging. The algorithm can do this now very effectively. And now what we have is a way to quantify how motile those immune cells are over time. Okay, so the boundary condition of a single cell is shown in the white dotted line, and each of these axes represent the motility of those immune cells between 6 to 10 days after the LPS injection. What we find is the motility is very high initially and then subsides over time as the inflammation resolves. The last point that I want to bring you into, and this relates to the second act of the talk, is that the mechanism by which those immune cells get there is actually really different as well. The blood flow increases in response to inflammation. We know this when we hit our thumb with a hammer, right? There's an increase in blood flow to that tissue. The same thing happens in the retina. But what we found in 2020 in this publication in eLife is that the way that arterioles and venules mediate that increase in blood flow is actually very different. What we found is that when you look at arterioles and track the blood flow, right? This is how we quantified the velocity of those single blood cells in the big vessels. At about 6 to 72 hours after that inflammatory event, the velocity in arterioles goes through the roof. It goes really, really high. But the diameter of those arteries are basically unchanged, whereas venules increase their diameter to increase more blood flow, but the velocity remains relatively constant. Here's another example of that. The artery, basically the same diameter throughout inflammation, whereas the venule increases its diameter substantially to increase blood flow. Why would biology do this? Well, remember, the immune cells have to get to the tissue. One way that they want to do that is they want to stick to the vascular endothelium. One way that a vessel could do that is to dilate, to facilitate more blood flow, but also decrease the shear stress or the velocity of those moving cells at the boundary condition. This would give a higher likelihood that those cells can bind and affiliate with the vascular endothelium in response to disease. Just a quantification of the same effect. Velocity is increased in arterioles, but remains constant in venules. Venules increase their diameter, whereas arterioles basically keep a very similar diameter, but the net effect of increasing blood flow is the same, right? The input and output are effectively the same, but the way that they achieve that increase in blood flow are through two very different mechanisms. So anyway, to summarize this talk, in the first part, we described how adaptive optics is a very important tool to image and quantify single blood cell flux at the level of the tiniest of capillaries, where disease initiates. It's also, in our opinion, a very effective tool to image the biggest of vessels and understand how vascular perfusion is changed in the biggest of vessels, and then be able to study from the biggest to smallest vessel in the same instrument, something that wasn't possible before. And then finally, combining this with detailed investigation with phase contrast, now we can reveal the behavior of immune cells, both with contrast agents and without, to image the motility of immune cells. We think this will be a very big deal for ophthalmology, because if we can track immune cells, now we have a readout which can gauge the inflammatory state of the retina beyond what OCT could show us before. Just the thickness of the retina only tells us about the local swelling. Here, the number of immune cells and their motility tells us about the cellular components that are mediating that information. I think that's time for today, but without opening up the floor to any questions, this is, of course, the team that makes this work possible. I'm here captaining the ship, but all of this crew is really making this work possible. This is talented graduate students in neuroscience, optics, and bioengineering, and also a small sea of technicians, optical engineers, and a team that really makes the science possible. So thanks for your attention, and happy to entertain any questions. Yes.
Mean and how does it apply to say drug delivery and uptake? Is it possible to view uptake long term, not just the information phase, but when the drug is effective and it's leaking outside?
That's
is there any way to?
Great. So let me repeat the question for everyone online. The question is, how is this applicable to drug discovery and drug evaluation in response to therapy? So we're very excited about this application for the following. I'll give two examples of the application. The first is at the capillary level. As many of you are familiar, that anti-vascular endothelial growth factor, anti-VEGF therapy, is one of the most common revolutionary drugs that have been given to people that suffer from diabetic retinopathy, wet form of age-related macular degeneration, et cetera. But the data to date is very puzzling, because sometimes you'll inject these agents, people show an improvement in vision, but the mechanism by which they improve vision is actually poorly understood at this point. We think adaptive optics, by imaging the capillaries, might shed some light on why these people are getting vision back, preserving vision, regaining a little bit of vision, by understanding the perfusion at the capillary level. So that's the first answer. The second is in the context of inflammation. So drug discovery in response to inflammatory cues is a hot topic right now. The central nervous system is, of course, immunoprivileged. That's the way that most people think about it. And so it's a particularly tough location for drugs to target. And it's also a pathway in which we can modulate immune cell extravasation into tissue. One of the more attractive features is to modulate selectin and integrin binding of these white blood cells. If I'm a drug company and I'm building a drug that can suppress the ability for those cells to bind to the capillary or the venule, we're going to limit their ability to get into the tissue. And that's exactly what you want to do in autoimmune disease. So autoimmune disease where an antigen is expressed incorrectly and the body starts attacking itself, you want to be able to suppress those selectins and integrins. And so we think this will be a great evaluation tool to see how effective those therapies are working, using the eye as a model for the entire systemic circulation. Great question.
There's a bunch of questions on Zoom, so I'm
Okay.
going to balance in-person questions versus Zoom questions. The first is, does the hematocrit of a particular capillary remain relatively constant or change rapidly?
It changes quite rapidly in our hands. Occasionally, oh, I'm sorry. The question was, does the local hematocrit of a single capillary remain constant or does it change rapidly over time? We found both conditions. In a paper that is in press at the moment, we quantify exactly that question. If we come back to the same capillary 10 minutes, 15 minutes later, an hour later, a week later, a month later, what does that look like in terms of its stability over time? Largely, single capillaries maintain their flux over time and their hematocrit over time. But if you look at the ultra short time epochs, you'll see these little bursts of these big boluses of erythrocytes and local hematocrit, which changes over time. So it's a little bit of both. On the local scale, it's incredibly variable, and over time, it's actually quite stable. Great question.
You can look up blood flow. How do you look at that? What do you know what the effect would be?
Great. So the question from the audience is, have we looked at blood flow in animal disease models? And I didn't present any of the data today, but be on the lookout for a publication in IOVS which addresses this. So our R01 currently is looking at, does blood flow change in the early stages of diabetic retinopathy? And for this, we're using mice that have a single point mutation in insulin, which allows it to accumulate in the pancreas. And basically they have a hyperglycemic state that looks just like uncontrolled human type 1 diabetes. In those mice, remarkably, here's the Cliff's notes on those findings. What we found was really pretty interesting. In the early stages of diabetic retinopathy, and at least in these mice, there was no changes in the hematocrit, the flux, the velocity, or total blood flow in those mice, which we found just remarkable that this was a null finding. Because when you look at the literature, all other indicators suggest that there are profound vascular changes in these mice in response to hyperglycemia. So again, we think that increased sensitivity here has potential to shed light on what those early mechanisms are that change blood flow in the microvessels of the mouse eye.
Another Zoom question here. Any sense whether some of the immune cells are resident within the retina instead of ones that extravasate from the vasculature?
Great. So the question is, how do we know the immune cells are ones that come in through the systemic circulation versus ones that are already there? And so there's a number of different immune cells that already exist within the retina. And the most popular or most studied are microglia. And in 2021, we had a publication which characterized, again, using transgenic mice, which cells were the native population and which cells were coming in. And in that publication, we studied the activity of microglial cells. So the short answer is yes, we can know which ones were already there within the tissue and which ones are new and that arrive locally within the tissue. We have a very interesting model that we're working on this year, which is called Adoptive Transfer. And this is again through a collaboration with Colin Chu. The experiment is pretty cool. You take one mouse that has fluorescent lymphocytes or any kind of immune cell that you might care about. In this case, we're adopting CD4 positive T cells. We're taking those cells out of one mouse, we're breeding them up in a dish in vitro. We're purifying that population, and then we're injecting that fluorescent population to a second mouse. Why is that applicable to the question? Because now we know exactly not only which cells are arriving to the tissue, but what their origin was as well. They were injected into the systemic circulation, they're arriving to the host animal from a different fluorescent mouse. Yes.
So thanks for a great talk. Obviously, wonderful technology. How far are you from the point of taking these different changes in blood flow and matching them with electrophysiology and how the nerve cells and photoreceptors are reacting to the changes in blood flow.
Yeah, so the question is, how can we relate some of these blood flow metrics to more conventional ways of maybe electrophysiology at the single cell level, or maybe multi-electrode arrays or ERG? We haven't done a lot of this today. We think it's a really important question because if blood flow can serve as a surrogate for electrophysiology, this would be really important. It's sort of an empty answer. I think it's important. We haven't really made those connections yet. So that's area ripe for investigation.
Do the red blood cells in contact with the lumen change shape or speed?
The question is, do the red blood cells in contact with the vessel lumen change their shape or speed?
Yes.
Yeah, so I just want to first make a little bit of a correction there, I think, in the question. So most of the cells that we see that are coming in contact are white blood cells. Those are the rolling cells, at least. In capillaries, maybe the question is in response to capillaries, the rheology of those cells do change as they interact with the capillary endothelium. So I won't bring up this slide, but maybe you recall there was a slide of the different packing density of those blood cells. One of the most interesting targets that we think is untapped in understanding microvascular disease is the shape and the rheology of those blood cells as they squeeze through the capillaries. Several diseases in ophthalmology are known to affect the shape of those red blood cells. So for example, sickle cell retinopathy is a disease that affects primarily the African-American population that have basically a sickle-shaped red blood cell. Instead of that biconcave shape, they change their shape. And what you see in sickle cell patients, this is work done by Richard Rosen and a number of his collaborators, have found that you can actually image the aggregations of those sickle cells within capillaries, again, describing the mechanism by which you might get that retinopathy inside a living human eye. So I hope I addressed that question about red blood cells and their deformation in capillaries.
I have another Zoom here. Any thoughts on what mediates the differences in flow morphology, i.e. stacked or chaotic, etc.? Would anticoagulation alter this flow morphology?
That's a great question. So the question is, can we change the flow morphology that we see in these capillaries by giving, for example, an anticoagulant? We haven't done that experiment yet, but an anticoagulant in particular is thought to primarily affect the platelets and the aggregation of microthrombi that form within these capillaries. And so in conditions where we do see aggregations of platelets that create these clots, an anticoagulant would be really interesting to study. We haven't looked at that yet.
So early on, you showed the capillary, some of the capillaries arrest the blood flow briefly. And you commented that that turns out to be more common than we thought. How long do they arrest for, and how frequent is it?
So the question is, how often do single capillaries arrest their blood flow, given that this was sort of a rare phenomenon? Let me start by sort of giving sort of the dogma or the textbook version. In the central nervous system, it's generally thought that all capillaries are perfused all the time in a healthy brain and healthy retina. In other tissues, that's not true, right? In like skeletal muscle tissue and other organs, there's what's called capillary recruitment. So you recruit more capillaries to become perfused when you need more blood flow. That generally is not thought to happen in the central nervous system. The second part of the question was, how long do those capillaries remain occluded? This is really what caught us by surprise. When you look at those otherwise healthy capillaries, sometimes they'd be occluded for minutes at a time. And that's a long time for a capillary to become occluded. In the context of stroke, we all know that it doesn't take very long for populations of neurons to be lost after maybe about just 4 minutes of having no perfusion. So the fact that we were seeing this for minutes at a time was very long for those to be non-perfused. I think there's a ton of interesting questions here for senescence and aging, because as more and more people get interested in cognitive decline and Alzheimer's disease, there's a hypothesis that the number of capillaries or the vascular perfusion at the microvascular level might be compromised in a lot of those neurocognitive decline diseases. It'll be interesting to see if the rate of occurrence of those stalls increases in conditions of Alzheimer's. And there's a sea of researchers that are tackling just that question in 2-photon imaging of the cortex. It would be really interesting to see if the eye, again, could serve as a surrogate marker to see what's happening in the brain by looking at the microvascular perfusion in the eye.
for the pro-degenerative eye equivalent of glaucoma.
Absolutely.
Any other questions? We've, I think, exhausted the list on Zoom as well. So, all right. Well, with that, let's thank Jesse one more time. Truly fascinating, fascinating topic and just fabulous images. To all of you that have joined us on Zoom, thank you so much. And this will, I guess, conclude our seminar for today. So thanks again.